Good evening. This is my first time ever in Marin for a book tour, and I'm pleased to help support independent bookstores. Book reading is still based on browsing, at least for the age of this average group. It's harder to browse online, so I like independent bookstores. But I'm going to tell you about Life at the Speed of Light. It's sort of in three parts. The first part is the history of this field, mostly for the last 70 years. But in fact, on researching the history, it goes back to Aristotle, and you think you've come up with a novel idea, and then you find a Frenchman wrote a book in 1901 about synthetic life and how they were so certain somebody would develop synthetic life one day that the world should not be the least bit surprised when it happens, because it's so obvious that it will occur. It's probably a good thing I didn't read the book before. I wouldn't have done it. But people have had these ideas, these mechanistic views of life, long before we knew what the actual life components were. In fact, the book starts out, and it's based in part on a lecture I gave last summer. I was asked to go to Dublin to Trinity College, to the very same site where Schrödinger gave a key lecture in 1943 that turned into a book, a very small book that he wrote. So some of you, I hope, have seen it. It's called What is Life? And it's influenced generations of scientists, notably Watson and Crick. I was very influenced by it. And Schrödinger, as a physicist who was asking questions, does biology obey the laws of physics? He didn't know much about biology, so he dove into it to see if there was something special about biology, or was it subject to the same laws that the rest of the universe was. And he largely concluded it was based on the same rules. But thinking about things, in 1944, when his book was published, the scientific community was certain that proteins were the basis of passing on genetic information. That DNA was way too simple of a polymer, and they thought it was just some sort of support matrix for the proteins that carried all the information. But he surmised that whatever the genetic basis of inheritance was, it could be remarkably simple and still carry tremendous amounts of information. He proposed it could actually be as simple as the Morse code with dots and dashes. And so his book inspired Watson and Crick to actually try and find out the structure of the inherited material. But in 1944, a key experiment was done. It was the first one to prove that DNA was actually the genetic material, not protein. But it took a long time for this to catch on, and even when Watson and Crick published the structure of DNA in 1953, a lot of people took notice of their paper because they were still so certain that proteins were the genetic material. It wasn't really until the 1960s when Marshall Nirenberg and Goban Khorana actually worked out how the genetic code codes for proteins, and then it was triplet sets of base pairs that coded for each amino acid. And Nirenberg did this. He actually used synthetic DNA for the first time, and used an in vitro protein synthesis system, which means he had all the functions of the cell without having a cell wall. And they just made synthetic DNA and fed it to it and see what proteins were produced. So it wasn't until the 1960s then that people really started to believe that DNA was the genetic material. By the 1970s, the world was changing very rapidly. My friend and colleague Ham Smith discovered restriction enzymes. These are the molecular scissors that cut DNA at very precise points. And this led to the development of the whole field of cloning and molecular biology. Also in the 70s was when Conan Boyer did the first gene splicing experiments, and Stanford and UCSF patented this technology. And also the first genes of commercial interest, they patented the gene for human insulin, and Genentech was born to start producing human insulin. Until then, people with diabetes only had pig insulin that was isolated from slaughterhouses. and a lot of people who develop resistance to the pig insulin develop antibodies against it. And so human insulin is the first biotech product, totally changed the world of diabetes treatments. So it wasn't until the mid-70s, and so I got my PhD in 1975 at UC San Diego. The first genome was sequenced in 1976. It was a small RNA virus that was sequenced in Belgium. 1977 is when Fred Sanger sequenced the first bacteriophage genome, a small viral genome. And that was 18 years later that my team sequenced the first genome of a living organism. It was a bacteria with 1.8 million letters. It was only 5 years after that we jumped up to 3 billion letters of the human genome. So things have been changing dramatically. And we've been going in this short period of history, first learning what the genetic material was, and then by reading it, we've been what I've been called digitizing biology, as we take the 4 letters of the genetic code and sequence them and put them in computer databases. We're converting those 4 letters into the ones and zeros of the computer world. At the same time, all this was happening with DNA. Protein structures were being determined, and it became clearer and clearer to the community that DNA coded in a linear fashion for the linear protein, but contained in all that information was how they fold properly into the right shape. And you know, the number of possibilities, it could take 1 billion years to try all of them. But in each of our cells, in a matter of microseconds, proteins fold into exactly the right shape. And the energy that drives all this was first described in the early 1800s by a scientist named Brown, who was looking through the microscope and saw pollen grains jumping around under the microscope. And the thing he observed is called Brownian motion, but it wasn't until 75 years later, when Einstein worked out what he was observing, that we started to understand the energy that drives all the activity in the molecular world. And it's simply what Einstein showed. It was the molecules that form water above a certain temperature bounce around with very high energy. And so this energy drives the proteins folding in the proper way in a very rapid fashion. But proteins are very unstable. In all of your cells, your proteins have half-lives of minutes to hours. And all that's coded for in the same genetic information. In fact, the amino acid at the end of the protein determines how long it will live in your cell. So all this is built in to the DNA programming. So we actually wanted to try and prove all this in an absolute way. So we decided to start not with life, but with the digital code in the computer databases, and see if we could start there and recapitulate life. So we started in 2003 after we finished sequencing the human genome, which is described in detail in my first book, if you want to have the whole context for this. It's called The Life Decoded, so I had to get plugs for everything here and help bookstores survive. It's totally selfish. But the techniques for writing the genetic code are pretty primitive. Scientists have machines that write short stretches of roughly 20 letters. These are called oligonucleotides, and they're used for sequencing primers and for PCR. But nobody's ever been able to build big pieces of DNA, and it's in part because these machines that write the genetic code aren't very accurate. So it's called the N-1 rule. The longer you make the piece of DNA, the more errors there are. And so we had to develop techniques for correcting the errors in the synthesis. And we started with pieces that were 60 letters long, and we put them together, and we built the entire chromosome of this small bacteriophage, the same one that Sanger sequenced, called Phi-X174. And we had this piece of DNA that was 5,300 letters long, and we injected it into E. coli, the bacteria. And as soon as we put in this chemically made piece of software, the bacterial system started reading that software immediately. It started making the proteins coded for in this piece of software. The proteins self-assembled to form the virus, and after thousands of copies of the virus are made, the cells burst open and release it, and then the virus infects other cells around it. So we call this a situation where the software, this chemical piece of software, builds its own hardware. All we did was put in the information system, and out of it we got a biological system that can infect other cells and make copies. Our goal was to actually make an entire bacterial chromosome of around 1 million letters. So doing 5,000 letters was okay, at least prove we could make small pieces with high accuracy and move them up in a simple system. So the story took 7 years from that point to develop all the tools and techniques, and these are described in detail in the book. You know, everything doesn't happen in a nice, straightforward fashion. There's lots of stops and starts and blind alleys, and things work and things don't work. But we got so we could write the genetic code with bigger and bigger pieces and do it in a very accurate fashion. And we finally learned to build these really large chromosomes. And we reported the first one in 2008. It was the largest chemically made substance by humans of a defined structure. And so while the chemistry was improving, we had to work up how do you boot up a really large piece of DNA. And so we had a team working on how to transplant genomes into recipient cells. And this took several years to work out, but it led to one of the most important papers, and there's a whole chapter on it. By simply replacing the genome in a cell, we completely converted one species into another. And this was a surprise to a tremendous number of people in the scientific community, as well as obviously in the lay community. And because it's so important, you know, it's similar to what happened with the viral DNA, but we had to use all kinds of more elaborate techniques to move around large chromosomes, because DNA is very brittle. You can't just pipette it and move it around. We have to move it around in tiny gel blocks, use electric current to move things in and out of the blocks, and actually then to do the transplantation and get the chromosome to go into cell. We have an enzyme that dissolves the gel, just leaves the DNA to go in the cell. And there were lots of problems associated with doing this for the first time. But it was a very exciting moment when we were able to actually do the transplant. We take a genome out of one cell type, isolate it, and transplant it into another. And it created the original cell that the DNA came out of. There was not a single molecule left out of the recipient cell. So simply by changing the DNA software, you convert one species into another. It turns out this has happened many times throughout evolution. When we started sequencing genomes from organisms in the ocean, a lot of them had multiple chromosomes. And everybody thought that evolution happened by making small random changes, and then they would have a significant impact, and you would slowly get an evolved function. But what we found out is through these same kind of mechanisms in nature, sometimes there's a mismatch in the systems, and cells can merge together, or the DNA is taken up, and it adds entirely new functions, sometimes thousands of new functions in a single step. So evolution went on in some big steps. It didn't just go in this tiny little one mutation at a time. In fact, the entire plant world is based on a microbial cell, an algae cell, getting taken up in a eukaryotic cell, and that's how the chloroplast system developed. There was actually a bacteria that got engulfed and became the photosynthetic center of plants. And so the entire plant world is based on a bacterial cell being engulfed in another cell. Every one of our cells have mitochondria in them. Those are the energy plants, and I think you know that the mitochondria has its own genome. So you only get your mitochondrial genome from your mother because you start with the egg. And so that's one piece of inheritance that only goes from mother to child. And so you can trace lineages with this, but it's the energy plant in all of our cells, and that came from another type of bacteria. So evolution happened by some of these big dramatic changes. We were finding a way to do it naturally in the laboratory and make these rapid changes. So what is described then is all the process for then making this 1.1 million letter chromosome and activating it in this recipient cell and then creating the first synthetic cell. And that's what we reported in 2010, and it was an exciting moment for us with all the years of effort going into it. But it was a demonstration of all the principles that we had together, and we call it proof by synthesis. So the chemical world, there's probably 10,000 papers published a year with the part of the title being proof by synthesis. That's how, if you isolate a chemical substance and then you remake it and it has all the properties of it, that's called proof by synthesis. Only now we've done this with the entire software of a living organism. The third part of the book describes sort of what are the implications of this for the future, because now we can rapidly interchange the digital world and the genetic world. We can convert DNA sequence by reading it into the ones and zeros of the computer. And now we've shown we can go in the other direction. All we need is the computer code, 4 bottles of chemicals, and we can rewrite the genetic code and start to design organisms and change evolution at a very rapid rate. Some of the uses that we talk about in the book are, and where it got the title of Life at the Speed of Light, because we can interchange the biological world with the computer world now. Once it's in the digital world, we can send it as an electromagnetic wave or through the internet at the speed of light. And we have a conversion device now that can take that information at the other end and recreate biology. So whether you call it a replicator, we call it a digital biological converter, we refer to it as biological teleportation. It's all describing the same thing of being able to start with life, digitize it, send it as digital information, and regenerate it at the other end. And I talk about the problem of sample return from Mars. The problem is getting a rocket up there that's big enough that it could blast off from Mars and get back to Earth and not contaminate Earth. And there was a multi-billion dollar program for a long time dedicated to sample return. And finally they sort of gave up on it because it was too hard of a problem. And even for a long time, even with this discussion of colonizing Mars in the next couple of decades, they're talking about all the trips being one-way trips. They don't know how to get people back. So that's why it's going to happen in the private sector, not with the government. But we worked out using our biological teleporter, if we discovered DNA on Mars and sequenced it, we can send it back to Earth in as little as 4.3 minutes when Earth and Mars are very close to each other. On a more practical scale, all of you are aware of the flu virus. I don't know if any of you were old enough to have been around in 1918 when the flu killed 3% of the world's population. But not too long ago we had the H1N1 pandemic that killed 250,000 people in this country and came from infections in Mexico City that rapidly spread through airports and then affecting a lot of people. There was not a new vaccine until 2 months after the pandemic had passed. And if it was actually as bad as they predicted, that would have been a massive problem. So we've set up to use our new technology, working with BARDA and the US government and working with Novartis. Instead of taking months to develop a new vaccine, we can do it in less than 10 hours. And we do it just using our synthetic DNA tools where we write the genetic code. And more importantly, we can send it as digital information. So in fact, the test was BARDA sent us the H7N9 genome sequence, and they gave us less than 12 hours to make it. We did it in 10 hours and had it ready for Novartis to start scaling up. So we've actually built the first unit at Novartis's new vaccine factory in North Carolina, where they're, for the first time, they're using mammalian cells instead of chicken eggs. Normally we consume 900 million chicken eggs from making the flu vaccine each year. Vaccine production is finally entering into this century and using mammalian cells, so it's much faster with much higher yields. But we have the first digital converter in North Carolina, so as soon as there's a new pandemic, we can just email the sequence there, and in 10 hours they'll have things starting to go into production. This got a very real test in April when the H7N9 strain broke out and started causing a number of deaths in China. The Chinese sequenced the DNA, sequenced the strain right away, posted the sequence, and at the request of the government, we made the vaccine strain in 10 hours. And for a long time, our strain was the only source that the CDC had and the government had, because sending this highly infective strain through the mail was not something anybody really wanted to do. Mm-hmm. So these new technologies add a certain safety margin to things, but also we can now send a vaccine around the world in less than a second. And if you have one of these conversion devices, you can rapidly make the vaccine. So right now we just have the one test site at Novartis, but I talk in the book how I can envision maybe every country having one, every major city, perhaps schools, or even in the future having a box attached to your home computer where you can download biology from the internet. Mm-hmm. Right now we can download insulin from the internet. So instead of having it made at a company, we have a device, we'll just download and produce, the box will produce insulin. We can produce the vaccines, and we're working on bacteriophage as new antibiotics, because we now, for the first time since the discovery of penicillin, we have more people dying with drug-resistant bacterial infections than ever before. In fact, more people died this last year from infections than from automobile accidents. Mm-hmm. So we're entering into this sort of very dangerous phase. We're trying to use this technology to develop new ones, and we can actually send them around the world in less than a second with this new technology. We're thinking of hiring some 11-year-olds to tell us what the real uses will be, what biology we really want to download from the internet in the future. But I think these things just help illustrate what our teams have shown over this period of time, that the digital world and the biology worlds are becoming totally interchangeable, and that's going to be a big part of our future. I look forward to your questions. Do you want people to use microphones, or are you okay?
If you could just repeat the question.
Okay.
Craig? That'd be great.
Please, sir.
What are the security implications of what you're describing?
Yeah. So the question is, what are the security implications of what we're describing? So it's something that several have asked over the years while we're doing this. So, and I describe some of this at length in the book about all the ethical review and things that we've gone through. The first study was published in 1999, because it turns out ethicists have never considered the question of is it okay to design and make new life in the laboratory. And then the Sloan Foundation funded my institute along with MIT and a Washington Security Center to look at the bioterrorism and security risk. And you can download that multi-year study from jcvi.org. And when we announced our first synthetic life form in 2010, President Obama asked his new bioethics panel to take this on as their #1 challenge. And they published a report in December of that year that you can download from the White House website. And the long and short answer is, it's clearly a dual-use technology, like almost all new technologies. But there was less concern with it because you can basically go to any farm, and if there's a dead cow there, you can get anthrax from it. You don't have to try and synthesize it in a high-tech lab to do that. And a lot of other lethal agents that if people wanted to use them for bioterror, they could readily get their hands on them. I'm not sure I found that really comforting. But I guess the good news is terrorists more like things that go boom and attract attention than kill plants and animals and people, but I'm not sure I find that really comforting either. So, you know, basically the solution is pretty straightforward, and there's a far bigger threat from new emerging infections, like a new flu strain, or in October we passed 7 billion people on the planet. Not too far away, we're supposed to have 10 billion people. That's going to lead to all kinds of new diseases and outbreaks, and we need new antibiotics and new antivirals. We need those today with these new strains that are resistant to everything we have in our arsenal. And the solution is the same. It doesn't matter whether it's a bioterrorism agent or a new emerging infection. We need ways to counteract it, and the means are the same for both. Pharmaceutical companies aren't particularly interested in developing antimicrobials because they don't make much money on them. In fact, the ones that did in the past went the wrong way. They made things like Augmentin that worked broadly on almost every type of bacteria, because then they could make one drug and have a huge amount of sales of it. And that's taken us all in the wrong direction, because that just caused a wide range of bacteria to develop resistant to that one antibiotic. So whether it's a federally driven program or however this is going to happen, it's not hard to come up with new antibiotics. It's hard, I guess, for companies to make enough money on them when they target just meth-resistant staph or something. So, you know, it's a problem we have to solve nationally. We can't expect one industry just to solve it for us on its own. That's probably more information than you wanted. Yes, sir.
I'm curious, there's a lot of anti-GMO, genetically modified organisms. Wouldn't it be great if we could design a plant that could grow in arid regions that basically fulfill the minimum daily requirements, the dietary requirements, and you could design a plant that would grow in certain, let's say, areas of Africa where, and that would provide basically a food source that would provide a complete dietary program for the people.
So I totally agree with you, and we're actually working on some, and we're talking about the possibility of creating this new algae flower in India. India has basically a population equivalent to the size of US that is the bottom of the poverty and nourishment scale for the whole world. And it's not hard to do. It's the social acceptance of this stuff. It's the people that are actually starving to death that are the most concerned with some of these notions. And another author that I was on a panel with in Boston said she did a survey and found that 50% of people don't know that tomatoes have DNA in them. And if that's where you're trying to start a conversation, it's pretty difficult because our fundamental knowledge and training in this country, a sophisticated country in theory, is so poor it's hard to have an intelligent conversation. There are things out there. The golden rice actually in Africa, in India, could actually prevent easily 250,000 cases of blindness in kids each year. But the Europeans are blocking it from being grown in Africa because overfed Europeans don't like GMOs. And so we're not lacking in science breakthroughs in this area. We're lacking in science understanding and social acceptance. So it's not hard scientifically to achieve what you're suggesting. And a lot of people are thinking along those lines. How do you overcome this blind ignorance, even amongst the people that would be most benefited by having nutritious food sources? Yes?
I'm involved in the brain cancer research area, and there's been a lot of advances in vaccines that are used genetically to target or help prevent cancer. Are these something that that your technique might end up being able to create?
So the question that probably couldn't hear in the back, it's more around cancer vaccines. She works on cancer vaccines and what our techniques help with those. And we're assuming the answer is yes, because you can basically rewrite any genetic code and have it target things. cancer vaccines are kind of a mixed bag, and nobody really knows why they work, and sometimes they work and sometimes they don't. The latest things that look very promising are the immune system stimulators that just activate our entire immune system and help knock out cancers. And a lot of data shows that when people get cancers, it's quite often periods of stress or when their immune system is partially compromised. And so activating the immune system can go a long way. But all these areas are moving forward as we understand the human genome better and as we use these new tools. So I'm hoping the short answer is yes. Yes, sir.
I'm not quite how to phrase this, but is there more of a danger of genetic codes going out of control when you're introducing this artificial genetic code from what's happening naturally within the environment, and how do you control for this? How do you predict, et cetera?
Yeah. The short answer is you can design stretches of genetic code so they evolve and change very quickly, or they change much slower than your DNA is changing. And if we're actually making industrial organisms to produce food or medicines, we actually want them to change very slowly, and it's easy to control that. It's not easy to control your DNA and how it's changing, or when crops, all the things that we eat now that we consider natural foods are all the result of random blind genetic experiments. Natural occurring tomatoes are bitter green things about the size of my thumb. Red tomatoes that we think are so natural and great have all been genetically bred. And the reason all of our plant crops, they actually have very confused multiple copies of genomes in them from all the intermixing and interbreeding and random experiments that have been done. So everything we eat is pretty unnatural, and nobody knows the long-term effects of these things. I'd much rather eat a well genetically engineered crop any day, but we can control rates of evolution and how well they change. Yes, sir.
Somehow I got it in my head that your ultimate goal was to create life from scratch in the laboratory. Is that wrong, or is that...
So I spent a whole chapter trying. Nobody's sure what they mean when they say life from scratch.
Well, not using a sort of, in a sense, cheating the way you're doing it now. I'm not degrading at all.
Can you do it?
I don't spend every day thinking about it, but if I did...
Well, you should try it, then you can work out whether I'm cheating. We're not trying to recapitulate 3.5 billion years of evolution. There's a lot of labs doing it, and I talk about some of those in the book. Jack Sosaks at Harvard is trying to put RNA molecules into lipid vesicles to see if they'll start developing some of the properties of life. All that is really fascinating stuff, and may not tell us how life might have ultimately evolved, but could have easily come because somebody designed it in a computer on another planet, and it came here on an asteroid. It's more of an academic question than anything else. We're building on top of 3.5 billion years of evolution. Every species on this planet is a DNA software-driven species, you included. And if we change your DNA, we will change who you are and even what species you are. I don't think that's cheating. That's using advanced knowledge and intelligence. And we talk about this notion of building life from scratch, and I talk about making a cake from scratch. Most people think that's getting flour and eggs and sort of mixing it together versus getting a cake mix. That's making a cake from scratch. They don't talk about getting carbon and oxygen and making the basic elements and putting it together, right? And so, I'm not picking on you, but you volunteered. I don't think you have any idea what you mean by making life from scratch.
Well, I know a question I'd like to answer, which is, how probable is it that life got, started anywhere in the universe?
Well, it's pretty probable. We're here talking about it.
Well,
in fact, I'd say it's a certainty.
Is it like? I bet where's the net? Was it a weird thing, strange thing that happened on Earth?
No.
Once in a million times, or was it something that we're likely to see elsewhere?
There's a couple of bumper stickers I want to get. One is life happens. You put the right things together, we're going to find life everywhere in the universe. In fact, most life on this planet is not something you can even see. We live in a bacterial world. In fact, we discovered what's called the microbiome now. You have 100 trillion human cells, but you have roughly 200 trillion bacterial cells. Not just you, I don't think. Ladies next to you do too.
All kinds of people travel with me.
So, 10 pounds of your weight is just bacteria.
Not to mention the viral.
Yeah, the viruses sort of go with each bacteria, and they're even harder to track because there's 10 viruses for every bacteria. We have 22,000 human genes. We all have about 10 million bacterial genes. And in the air you're breathing, we're going to be publishing a paper shortly measuring the bacteria and the viruses in the air. During the course of the time in this crowd, you're breathing in about 10,000 bacteria, several of them brought by the people around you. And we can find life miles deep in the earth. In fact, there's as much life deep in the earth than there is in the entire surface of the earth. But just weighing the bacteria in the ocean, they weigh more than all the plants and animals, things that we see in the visible world. And so while people are looking for Martians walking around on Mars, I spent a fair amount of time on the book on this. If you go down a kilometer, you find ice. If you go deeper, you actually find liquid water. It looks like Mars had oceans on it at least 2 different periods. And we exchange 100 kilograms of material between Earth and Mars every year, from meteors going back and forth. In fact, statistically, you can't take a shovel of dirt on this planet without it having Martian soil in it. So whether the bacteria originally came from Mars and fertilize this planet, or they go the other way, we've been exchanging bacteria for billions of years with Mars and other places. And so it would be a much bigger scientific finding if we don't find any life or even evidence of prior life on Mars. But I'm certain we'll find it wherever we look in the universe. And now that with the Kepler project, we're discovering in our own galaxy 100,000 Earths and Earth-like planets in the Goldilocks zones. It's pretty self-centric to think this is the only place it's happened. But we're not trying to recapitulate that, but it's probably a more important mechanism of how it spread. In fact, these same bacteria have novel mechanisms, because just think whether you're eating a tomato or a fish or something else, and you became a fish because you ate their DNA. Well, that's what happens in the bacterial world. So the restriction enzymes that Ham Smith discovered and got the Nobel Prize for in 1978 are how organisms protect themselves from alien DNA coming in and taking them over. And so it's a key part of evolution when those things become a mismatch. But we live in this DNA world. We're a DNA-driven species. Every species on the planet is. And it's all about that software. Any cell in your body, if you take out the DNA, the cell will die. Any species on this planet, without the software to keep making new proteins, they die very quickly. So thank you for the question of being patient. Yes, ma'am.
What about slowing aging? We won't create a new person, we'll just slow what we have.
Good. Somehow that's, looking at the age of this group, I'm not surprised by that question. And, that's occurred to me personally. And so...
Hurry up.
Yeah. We are trying to pick up the pace for doing this. What we're trying to focus on is through understanding the genetic code in large numbers of people to find out what the changes are due to that lead to early aging. Most of the advances that we've seen have been simply from improved sanitation, improved health. Immunization is perhaps one of the most important things we've done to increase longer life in the population. But for these next stages, we have to understand the fundamental mechanisms in the cell that do this a little bit better. But I think it's getting much closer. It used to be extremely rare to find people over 90, and rarely over 100. and this is becoming much more commonplace. Fortunately, in my own case, my own mother just turned 90 this last year. But you know, most people start to get a little fragile around that time period, mostly from muscle and bone loss. And so there's some thoughts on simply by increasing activity and a few other changes that might become available soon, by increasing your muscle and bone mass and using your brain versus retiring, you'll increase your own healthy lifespan, whether you live longer or not. You know, as they say, if you go on one of these starvation diets, you don't actually live longer. It just feels like you do. We try somebody else first. Yes.
Didn't Schrodinger also say that he thought some biologists would find a simple way to make people live forever?
I don't remember that, but
You don't.
and thus far he's been wrong. But but more and more people are saying that, and you know, as we understand the code and what it codes for, there's no a priori system for things to run downhill. Somebody mentioned all the viruses in the environment earlier. One of the more interesting discoveries we found in the ocean is, you know, and it's a much more sinister view of life, but the viruses actually carry some of the key genes for the bacteria to survive, including all the photosynthetic systems. And what they do is when they infect the bacteria, they upgrade their software. So if you're a virus that's dependent on this life form to stay alive, you want to keep them happy and healthy, and so they constantly carry new software and upgrade their software. So it makes you wonder who's really in charge, and that we may just be carriers for bacteria and virus, and they want to keep us around for a while. But understanding these mechanisms and renewing and changing the software is a key part of long, healthy life and cells. So if we can learn those rules and apply them, there might be hope for all of us. So, yes, sir.
What's your current thoughts about algae?
Well, until you asked the question, I was having a current thought about algae, but we have teams working on algae in a number of different ways at Synthetic Genomics. Some to make antioxidants like astaxanthin, and producing large amounts of EPA and DHA, but also we're engineering them to produce the perfect balance component of proteins, so that it's the right balance of all the essential amino acids. So as I said, we could make an algae flour that's extremely nutritious and can be produced in a commercial environment with 10 to 100 times the efficiency of anything that can be done in agriculture. So there's been a lot of talk about fuels. That's further away, but also fuels. We can make a container this size with a new vaccine or a new drug to cure aging, and that would be worth $1 million or more. As food, it could be worth $1 or $2 per kilogram. If it's fuel just to burn, depending on where in the world you are, it's 20 to 50 cents. So using these same things, there's obviously environmental priorities on CO2, but coming up with more efficient ways to produce food solves lots of problems. Agriculture is extremely inefficient, and it should be one of our goals to basically eliminate it. So I'm bullish on algae. Yes, sir.
Can you envision a system of self-replicating organisms that's not DNA based? I mean, maybe we find it here, maybe we find it in outer space someplace.
Yeah, people have talked a lot about this forever. Nobody's ever found one. Silica is the closest to carbon chemically, you know, but carbon is pretty special. And so I, as a scientist, when I see the evidence for it, I can envision it. But there is no evidence for anything but carbon-based life.
It's a DNA, not carbon.
Well, they go together. DNA is based on carbon too. So you can't have DNA without carbon. It's all the same chemical system. Yes, sir.
Can you describe what this box is that takes digital code in and comes out with biology? I don't visualize how that can be.
Well, we call it a digital biological converter, and we don't have one that we could sell you yet, but we have one we're experimenting with in the lab. It just takes the digital code. It's the equivalent of your printer on your computer. We have cartridges with 4 bottles of chemicals, and those 4 bottles of chemicals basically write out the genetic code in the fashion determined by the computer code that's driving it. And then we have a separate robot in there that assembles that small pieces of genetic code into genomes, and other devices then that take those genomes and either make a vaccine out of it, use it to drive protein synthesis, or a variety of other directions. So it's about half the size of this stage and about this tall right now. So we'd like to shrink it down a little bit more and have it work faster.
Thank you.
Yes?
Once it does, that box does make a genome, is the further construction process of making something that's actually living and perhaps self-replicating, isn't that terribly difficult? On the horizon perhaps, but...
Well, we use a recipient cell, and we're trying to design a universal recipient cell that we can put any piece of DNA in and we'll boot it up. It's actually remarkably simple, and you can do it even without cells, and they're called in vitro translation systems. So a lot of proteins that are used as drugs are made this way. So it's all the components in the cell, and they work in a cell-free system, but those aren't self-replicating. They eventually run out of gas, or you have to keep adding chemical energy to them. But our system just uses, as he said, cheats. We use a recipient cell, and that reads the genetic code and immediately boots it up. So it's remarkably simple in practice once you know how to do it. Yes, sir.
Would it be possible to put the synthetic genome into say a mixture of enzymes and nutrients and have it make its own cell?
Yeah. So could people here in the back? Yeah. So and it's an experiment we are trying to do. So can you put the DNA just in a mixture with nutrients and the enzyme systems and eventually get a cell out of it? And the answer is probably nobody's done it yet. They've made viruses that way, but not cells. But it's a matter of just getting the right lipid envelope, and I think that will happen. And I think it's not quite life from scratch. That's more like building a Ferrari from a kit, where you buy the engine and all the components. But I still think it will be an important step, because all life as we know it on this planet derived from other cellular life. It's called the cellular rule of biology, and that's still true for everything we know about, including what we did with the synthetic cell. There's advantages if you could boot it up just from a protein and chemical mixture, because then you could store it on the shelf a long time. And I'm on this project with NASA and DARPA called the 1,000-Year Starship Project, and it's whether to actually make the Starship Enterprise. But the problem of having people be shielded for centuries of space travel, which is what it would take to get place, is just too hard to do. And so I suggested we just send digital information and boot them up when they eventually get to the other end, because then you just get a little tiny spaceship. So it'd be nice to be able to do that.
So do you think there's anything different between a cellular life force and life force that, is there something before that? That's not made of cells, that is pre-cellular life?
Well, viruses are not made of cells. Some of these look like hypodermic syringes. They have a capsule that holds the genetic code. They have legs where they can land on a cell, and they actually inject the DNA into the cell. So that's how viruses work. But viruses can only replicate if they can hijack a system to produce more of itself.
But you're still talking about something physical.
Yes.
So do you think there's something non-physical that predates that?
Yeah, Brownian motion is the only non-physical force that drives the energy of all these things happening. If you're looking for a spiritual answer,
well, you're saying only...
I think Einstein found it.
And do you disagree with that?
No, I think Einstein's right that it was Brownian motion that drove all these processes.
Can you say what Brownian motion is?
It's the molecular motion of all the molecules. Fundamentally, all the water molecules in your cells are vibrating so fast, it's the equivalent to a force 9 earthquake going on inside every one of your cells all the time. And that's the energy that drives all these biological processes.
Well,
It's not that mysterious.
Effectively heat.
But you
Well,
may,
heat drives Brownian motion, you're right.
okay, I don't know if that's, I don't agree with that necessarily.
That's
And
okay.
it seems, thank you, I appreciate that. And it seems very simplistic because there may be something that hasn't been measured yet. That you're basing your whole thesis on something that's measurable.
That's called science. And it has to exist, and you have to be able to measure it, and you can always evoke ghosts and mysteries and God and magic forces and things, but if you measure them, they don't exist to the scientific world. If you can't measure it, it's not part of...
Then why, but they keep trying to do that. They're going to quarks and subquarks and...
Those are measurable.
Right, but they're still trying to get something below that, and so we don't know if it's...
Philosophy
at this point. The decision is out yet.
Keep hoping. Yes, sir.
First of all, I appreciate you coming here and talking to us. It's a real honor.
Thank you.
I saw you on Charlie Rose last week and got very excited when I saw that you were coming. So
That was part of this same tour.
Yeah.
2 days ago.
So one of the things that's come up, I grew up in an era, as you did, where the U.S. was one of the leading scientific nations, and now you see almost like there's a pure science that's still going on, and there's kind of like an anti-science culture. I have talked to people that post on Facebook that they went to the Creation Museum or something like that. How do you...
It's interesting, they use Facebook.
Yeah. Well, how do you kind of, you see things from a different perspective. How do you see kind of like this science development and anti-science development kind of working out?
Well, it's probably one of the most dangerous things affecting the future of our country, because we're a country that's based on innovation and breakthroughs, and we're a country and we are a society that is 100% based on science for its future. It's not a luxury anymore. There's 3 times as many people on the planet than in the year that I was born, in 1946. 3 times as many. And pretty soon there's going to be 5 times as many. You can't look backward at what your ancestors did and how they survived. We're going to have to find a way to survive with 5 times as many people as we had in 60 years. And we're not ready for it. We're not ready for it sociologically, psychologically. We don't have the food, we don't have the energy, we don't have the resources. And so if we're going to survive as an independent country that continues to thrive in the way it has in the past, we need to overcome this anti-intellectualism pretty quickly that's really splitting the country in two. The trouble is, as things get more complex in our lives, a natural solution for some is to turn it all off and resort to fundamentalism. It's happening in Islam, it's happened in all kinds of places, it's happening in the U.S. In an extensive fashion. I don't know how to change it other than to help show people that our future survival is based on new breakthroughs, new science, and new ways to cope with the way we have been living, because we can't extend that into the future. If you have an answer for it, I would love to hear it. Yes, ma'am.
I just think all of us are so impressed and inspired by the fact that you chose 2 really very lifetime kinds of goals, decoding life and then, finding life at the speed of light. What's next?
That's not enough.
Everybody writes a trilogy.
Well, I was the lady in the back had the right idea, so I told my publisher that I wanted to do another book, and they said about what I said, a life extended. So But I have to do it before I write the book. So. Yes, sir. But thank you for your comments.
I appreciate your comments on two things that I've read. One about you, which is that your group is working on microbes that eat coal. And the other being an article I read about IBM's application of Watson in other areas, particularly healthcare. And I wonder if the capabilities of a Watson can help accelerate what you're talking about.
So in the first one, we did have a major program. It was actually funded by BP to find the microbes deep in the earth that do actually metabolize coal and make methane. That's why there's been the search for methane on Mars. It is an indication of life. But you can get methane from two sources. You can get it biologically from these microbes that do, or you can get it from chemical breakdown. And it looks like the methane they found so far on Mars is chemically based, not microbial based. But the problem was, even though this was showing immense promise for a while, as soon as cracking to produce natural gas really started taking off, that's what shut down many of the approaches to create biofuels cheaply. And this is like the third or fourth cycle we've had as a country where there'll be an energy crisis. We try to find alternative sources, and oil prices get dropped for one reason or another, and we wipe out whole industries again. So, and that's what's happened again. Only the positive thing is natural gas burning is far better than burning coal and oil. It's still taking carbon out of the ground and burning it, but it converts readily. Natural gas is basically just methane, and when you burn it, it gets converted to CO2. So that's better than all the things that come out of burning oil and coal. And the other good thing is all these resources are in the U.S. You think it's good unless it's in your neighborhood. But it helps with energy independence. But it's kind of cut the bottom out of biofuels as alternatives right now until the efficiency of new organisms is so high that it doesn't matter what the cost of oil is, or there's a carbon tax to get us there.
And Watson, is Watson?
And Watson, so Watson is indicative of creating massively parallel computers. So in my first book, I talked about in 1999, we had to build the third largest computer just to deal with the data to assemble the first human genome. So the future of biology and medicine is going to be massive computing. Watson's an example of that type of approach. But if we're going to sequence 10 or 20,000 or 100,000 human genomes now, that's going to require massive computes, massive data storage. But the goal is to get it in within the next decade. But the most important thing you can know about your life is your genetic code. Right now, it's not that useful to know it. But there are some things where it is critical. If you have lung cancer, the most important thing you can know right now is your genetic code. Because 3 to 5% of the population have one letter change. And if you have that letter change, Pfizer has a drug that has better than a 60% chance of curing your lung cancer. If you don't have that one letter change, it's not so good. But that's indications of the kinds of discoveries that are starting to come out of the genome. If we get 10,000 of those, it will start to really impact life.
Maybe one more?
Yeah, you haven't asked a question yet, sir. So.
Well, with the capability to read and then write, the next logical thing is to edit or error correct. And I'm wondering, is it part of your efforts to look into cleaning up single gene problems, or even maybe developing like a cleaner DNA that could be reintroduced into humans?
So I like this audience. You've asked like really great, insightful questions. Maybe you listened to what I said before or something, but that's exactly what we are trying to do. So I've had stem cells made from a skin biopsy, and what we showed is that as soon as you take the stem cells out of the human body, the genetic code starts to degenerate relatively rapidly because it's not under the cellular control of being in the right environment. It's like taking a human out of an environment and put him in isolation. It doesn't work quite as well. And so we're trying to use our DNA tools, very much like you described, to correct the errors that start to occur in stem cells before giving them back as therapy. And if we can do it with the errors that are occurring as a result of taking them out of the body, it doesn't matter whether they're embryonic stem cells or iPS stem cells, the same thing happens. we could, in theory, make younger stem cells and use those for therapy. Because some of the hints are the number of people that have had bone marrow transplants from storing early umbilical cord stem cells. One of the interesting side effects has been a more youthful cellular activity across these people's entire systems. And so it may be possible to do what you're asking, you know, if we're limiting errors and even correcting things that were naturally occurring errors, we may be able to at least partially restore a more youthful state. Trust me, we're working on this as fast as I took out time for the book tour.
Back to work.
Across the whole genome, we give an estimate, the current best estimate of how much junk could be produced, like of all the base pairs, like if you were to compress DNA to just the important parts that would be needed for replication.